Hello. So we are at the end of week 2, and I don't know about you, but I have felt like this week has gone by very, very quickly. Um, but here we are. We're at the end of 2 weeks. That's pretty wild. I hope you're all doing really well, and it's been really great to hear from those of you that I've been able to connect with over Zoom. Would love to, hear from all of you and know how you're doing and get to see your faces. If you haven't seen these already, my guess is most of you have. Your quiz 1 and journal are going to be due by the end of this week. I think in general, we're going to try to be fairly flexible with due dates and timelines. Just thinking that, everyone's got different stuff going on in their homes, and this is impacting folks really differently. And of course, time zones too. So, I'm aiming for about 48-hour turnaround for most assignments, but certainly by the end of the week. And so if at any point, these deadlines don't feel achievable, you feel like you need an extension, just let me know. I say that also not just because I want to, hopefully, have you learn a little bit of biology by the end of this, but also because this is, my way to know that you're okay and you're, presumably all right, and that you're getting the work in because things are presumably chill where you are. If you didn't see the announcement yet, or if you have, I think the goal from here on is going to be to have your daily checklist posted by Sunday the week before, and have that so that at least you have a plan, going into the upcoming week, and that you can work that around your schedules, hopefully a little bit easier. Some of you have asked if that means that these videos will be uploaded at the beginning of the week also, and I think that as of right now, that doesn't feel like a goal that is totally achievable for me. That may change in the future as we go into these next couple of weeks, but for right now, I think that we're going to continue with this goal of getting things posted, the night before class, so that folks in all time zones can access them at least on time for when your normal class would be. So I told you that this video would be a little bit longer, and it will be. So I'm just going to quickly sort of go through the topics that I sent you external content for. So I had you guys learn about enzymes and sort of the biochemistry of that. And so, this is going to be, if you've already watched those other videos that I've posted, this is going to be just a review, but maybe in Miss Finney's words. So if maybe that's helpful for you, great. And, if, you feel like you've got a really solid handle on it, feel free to skip ahead to the new stuff also. So, where we left off the last time I did one of these videos was we talked about photosynthesis and this concept of feeders. And so we spent some time learning about the producers in a food web. And so the producers are our plants, right? And we call them this because they're able to create their own food in the form of glucose through the reactions of photosynthesis. And so we also have the heterotrophs, which is us, what we're going to be focusing on today. We have the heterotrophs, which are organisms that are unable to photosynthesize, so we're not able to produce our own food, and therefore are forced to consume other organisms to get the energy that powers our metabolic processes. That process is called cellular respiration, and we'll be talking about it probably next week. We also have the decomposers, who again, their job is to sort of recycle and break down biotic matter. So this is going to be anything that either is currently or was alive or is the byproduct of a living thing. And so, an energy system, an ecosystem, and the movement of energy through it sort of looks like this. It has different levels, right? And it starts with an input of energy from the sun. We have different levels, right? And then we have a little bit of energy that is lost sort of off to the side at the ascension of each level. We also have the loss of matter, which we'll talk about in just a moment. And that loss of matter at the change of each level, I'm just going to change quickly to my laser pointer, that loss of matter at each level as ascension goes off to the decomposers, where it's then sort of recycled and broken down into its molecular constituents. So the molecules that make it up, and then get recycled to the primary producers at the bottom. Now, these primary producers are plants, and they are at the bottom. They make up this foundation because of their ability to photosynthesize. And so, you know, the other reason that they're at the bottom is because, or I guess I should say, why photosynthesizers are at the bottom is because they are the only organism that is capable of accepting, processing, and transforming that input of energy that initially comes from the sun. These other guys can't do that. And so what's going to happen is every organism in the levels as they ascend are going to eat the level below it. Seriously? This is very different working conditions from Andover. Anyway, here we go. So photosynthesizers are, again, able to take energy from the sun and transmute that into usable chemical energy in the form of glucose. And so we define energy as the ability to do work, and matter as anything which takes up space and has mass. So mass is a unit of matter, and matter, again, is anything that takes up space and has mass. So your like physical body would be matter. And then the processes that power it are producing energy. So this unit that is able to do work. And so we have these laws that are actually more like laws of physics that we call conservation of energy and conservation of matter. So these are pretty famous. You may have heard of them before. So conservation of energy law tells us that energy is never created or destroyed as it moves through a system. Rather, it just changes forms. And now similarly, this is true for matter. So conservation of matter tells us that matter is not created or destroyed. Rather, it just changes forms as it moves through a system. And so we're witnessing this here in this mapping of the energy pyramid. So we have the input of energy from the sun, which goes to the producers. And now the producers have access to 100% of the available energy, the available cellular energy within this ecosystem. And so in order for the organisms that eat the plants, these primary consumers, these are our herbivores, in order for them to access that, they need to eat the plants. Now, you'll notice that this number changes to 10%. So what this is telling us is that as we go, you know, in the process of consumption of these organisms, which had access to 100% of the available cellular energy, we lost 90% of that energy sort of off to the side as heat. And so what was left for the primary consumers, these herbivores, is just 10% of the total available energy that was available to power an organism in this environment. And that number decreases by 90% each time we ascend. And so the secondary consumers, these are our carnivores, because they are eating the primary consumers. They now have access to just 1% of the total available energy with this system. And so by the time we get to the apex predators, which, to be fair, nothing is hunting them, they're at the very top of the food chain, they have access to just 0.01% of the total available energy that exists within this system. The other 0.99% or the other 99%, 99.9 I don't know, something like that. The other largest amount has been lost to the side as heat. And then again, all remaining waste matter we would be sent off to the side to be broken down and recycled by the decomposers. And so none of this would be possible without enzymes. And so we've been talking about metabolism a little bit. We've sort of defined what that means. And we talked about photosynthesis, where the most important enzyme involved in that process was rubisco. And we saw this in the light-independent reactions of the Calvin cycle. And so enzymes, you know, we've said before, are a special type of protein whose job it is to facilitate a chemical reaction. So the terminology that we use for that is catalyze. So their job is to catalyze a chemical reaction. And in the process of doing this, they are actually conserved. So this is really interesting because an enzyme is going to hop into a chemical reaction and it's going to help it happen a little bit easier. And then as it's helping this chemical reaction occur, it's not going to get used up. It can continue, it can go right back to the start and be reused again. And so the way that they catalyze these reactions, the way that they help them along, is by lowering what we call the activation energy of a chemical reaction. And so if we think of the activation energy as being the sort of push that a chemical reaction needs in order to begin, you know, in order to get started, we would say that the enzyme sort of like lowers the amount of push that's needed. It's like the enzyme is contributing something that makes it so that there is less energy required to begin that chemical reaction. And so the output of this chemical reaction in terms of energy can be either endothermic or exothermic. So look at our root words here. So we have endo, meaning inside, thermic, meaning related to heat. And then exo, relating to the outside, think like exoskeleton, and then thermic, again, related to heat. But we're not exclusively talking about heat here. We're also just sort of loosely talking about energy. And so an endothermic reaction would be one that absorbs energy or absorbs heat at the end of when it's happening. Once that reaction has occurred, it's going to absorb heat or energy in the form of heat. An exothermic reaction is maybe a little bit easier to conceptualize because it's going to put out heat or release energy as it occurs. So we can more sort of easily visualize this as like an explosion, right? Like that's sort of your classic exothermic reaction, something that is expelling a great deal of heat and energy once that chemical reaction has occurred. And so looking at this graph here, this figure, I'd like you to, as you're watching this, sort of pause for a moment and take a look and try to interpret it yourself. But once you've done that, we'll just see here that on this axis, we've got energy. And we're saying that on the opposite axis, this is showing us our reaction coordinates. So what they're showing us in here with these lines is a chemical reaction. Specifically, it looks like they're telling us that this is the reaction between carbon dioxide and water. And so with the product of that reaction, so you react these two together, you get a product of H2CO3. And so what we're showing here is that when we don't use the enzyme, when there's no enzyme involved in this chemical reaction, the activation energy is quite high. It requires quite a large push in order to get going, in order to produce this product. But if we look at the blue line, this is showing the reaction occurring with the help of the enzyme. When we include the enzyme in that reaction, that push that's needed is much, much, much lower. There's less energy required. There's less of an energy input required to begin that reaction. Now, this reaction, it looks like, would be an it looks like would be an endothermic reaction, because if we say that this is our starting point, this would be 0. You know, this isn't labeled, but I think we can rightfully assume that this is 0. And so if this line is dipping below 0, that indicates an absorption of energy, which would tell us that this is likely to be an endothermic reaction. And so the way enzymes function is actually pretty interesting. So we've talked quite a bit about how proteins are entirely functionally dependent on their shape and structure. So meaning the shape they have is what decides how they perform. And we've also learned about how that shape can be really influenced by its environment, but more on that later. Now, the way an enzyme reacts, I guess in a reaction, the way an enzyme is going to react within a reaction is by bringing together molecules, bringing together these reactants by binding them to itself. So we would call, in this case, the molecules that the enzyme acts on, we would say those are the enzyme substrate. And the enzyme is specific to its substrate. So you can't just throw any old enzyme into any old reaction and expect it to efficiently catalyze that reaction. Enzymes are pretty specific, and they're going to work for specific substrates. And so the substrates that it will work for are going to bind very specifically to the enzyme molecule in a mechanism that we call the induced fit model, which means that the charges of the enzyme are complementary to the substrate, and such that when the substrate binds to the enzyme, that enzyme will then sort of envelop it a little bit. It's not going to completely change its shape because that would modify its function, and we know that can't happen, but it does sort of mold around the substrate a little bit to more accurately and completely bind those reactants. And so the space where they get bound to is what we call the enzyme's active site. So they bind into there, and then when they've bound together, when the enzyme is working on those reactants, bringing them together so that they can react, we call that the enzyme substrate complex. And then once they've reacted, that product can be released from the enzyme's active site, and the enzyme can go back and do it all over again if it wants. It doesn't get broken down or changed or used up in the process of this reaction, or any reaction. That's really important. And so, you know, we said that enzymatic activity is really specific, and we knew that proteins are really influenced by their environment. So what I mean by environment is things like pH, things like heat, right? We know that those are things that can denature proteins. And if we denature a protein, remember what that means is that we are changing the conformation or the shape of the protein so that it no longer functions the way it is supposed to. So this is real bad. We don't really want that to happen. And so it's important that enzymes in the body, you know, where they are sort of floating around, those conditions have to maintain have to be homeostatic. They have to be maintained and in a really steady state, because the location of them is where they are best suited for. So for example, an enzyme that lives in the blood, you know, has to be maintained at is in the blood because it works best at the pH that the blood is maintained at, which is, you know, slightly above neutral. An enzyme that works in the stomach is in the stomach because it happens to work well at that pH. And so if the pH of the stomach changes, that enzyme will not work as well any longer. And so there's also this concept of reactivity of enzymes and their engagement with their substrates being really impacted by temperature. So we can increase the temperature and actually increase the speed of the reaction a little bit. Because when we increase temperature, what you're doing is increasing the movement of molecules. And so think about it, if things are moving around quite a lot, they're more likely to bump into each other. And if the reactants are, and if reactants that are capable of reacting, and we've got an enzyme that is capable of working on that substrate hanging out, then what we have is a situation where things are bumping up together all the more, and so your reaction is going to occur at a faster rate. The opposite is also true. You can influence the activity of an enzyme-mediated reaction, so a chemical reaction that is influenced and regulated by the presence of an enzyme. You can sort of modulate and regulate this by changing the temperature. So you raise the temperature, the reaction speed increases. You lower the temperature and the reaction speed can actually slow down. Now you can go too hard in either direction for this. You don't want to increase the temperature too much because you may denature the protein. You also don't want to decrease the temperature too much because the reaction may just stop. And that may or may not be your goal. There's also other ways to influence the reactivity of enzymes. So we have a class of molecules that's called inhibitors. And so an inhibitor molecule is one that stops the function of an enzyme in a couple of different ways. So we could have a molecule that is a competitive inhibitor, meaning that it fits directly into the active site of the enzyme in such a way that it blocks the substrate from binding to it. We could also have what we'd call an allosteric or non-competitive inhibitor. And what this does is, this is this guy over here. And so these allosteric inhibitors, these non-competitive inhibitors, are going to bind to a place on the enzyme that is not the active site. So it can bind what's usually called the allosteric site. But it could be other places as well. And so this allosteric inhibitor is going to bind to the allosteric site. And what this does is this changes the shape of the enzyme. So now we have, we've sort of messed with the charges of the molecule, and we may have changed potentially the shape of the active site. Now that substrate is no longer capable of binding to the active site that it connects with. Both of these mechanisms are actually kind of common ways that a lot of prescription drugs work. So certain medications that we can take will actually function by blocking an enzyme in the body. So there are neurotransmitter inhibitors that can be used in mood disorders, and a few others that are escaping me at the moment, but I will post those for your interest because it is a pretty fascinating class of drug and directly related to this. So we talked about enzymes. Those are our protein biomolecules. We also have carbohydrates that we really care about. And carbohydrates, their main job is to be stored chemical energy. So we talked about how the plant is going to create these sugars through photosynthesis and then use those to create energy. Carbohydrates are what we'd call an organic compound. So organic in this case has nothing to do with farming practices. It only has to do with whether the molecule is carbon-based. And so we'd say that any molecule that is made up of mostly carbon is an organic molecule. Conversely, a molecule that is made up of mostly other stuff, not carbon, we would say is an inorganic molecule. Most biomolecules are going to be, actually I would say probably all biomolecules are going to be carbon-based, with maybe the exception of a few molecules that are used in the body to sort of help along other processes. But all of the biomolecules that we'll learn about, so proteins, fats, carbohydrates, nucleic acids, are all carbon-based and therefore organic. And so carbohydrates are, you know, different sized molecules. They come in all different shapes and sizes. And these are composed of smaller carbohydrate monomers that create larger polymeric molecules. So we talked about this last term. So remember, the monomer is the single unit that can be sort of added to create a polymer. So remember, mono meaning one, poly meaning many. And so a polymer is many units of a monomer connected. And if we're looking at, you know, nomenclature, when we're looking at if we're trying to figure out if we've got a carbohydrate, we would look for this ending "-ose, so indicating it is a sugar, or we would look for this ending saccharide, which we've probably heard before, but is new for us in this class. But we have seen "-ose before, so we know that anytime we see "-ose or saccharide, we are talking about a sugar or a carbohydrate. And so the one that we're really interested in here is this guy, glucose. So sugars can come as, they can be monosaccharides, disaccharides, polysaccharides, you know, meaning one saccharide molecule, two saccharide molecules, many saccharide molecules, respectively. And so glucose has this formula, this C6H12O6. And so this is going to be our product of photosynthesis. This is the one that we really care about. And then we've got here, I'll move me a little bit so you can see that a little bit better. So we've got here our primary structure of the molecule. Remember, our primary structures are the linear just layout, the linear, like two-dimensional layout of a molecule. This here would be our secondary structure of glucose. So this is a way of drawing molecules. This is a mechanism of diagramming molecules that shows us bond angles. So this would be accounting for the different bond angles, like in a secondary structure. These dashed ones are showing that it's sort of set back, and this filled in wedge shows that it's set forward. And so this here would be our tertiary structure, because this is showing the full three-dimensional molecule that we have with glucose. And so in thinking about all of these molecules, right, we talked about photosynthesis, and we're going to be looking at digestion shortly. So this really brings into question, you know, what are different, you know, what is the terminology that we would use for different types of metabolic reactions. And so the two main ones that we have been learning about and will continue to learn about are catabolic reactions and anabolic reactions. So catabolic reactions are ones that break down molecules and create energy. So there's energy yielded from the breakdown of that molecule. An anabolic reaction is the exact opposite. So this is a molecule that this is a reaction that is creating a new molecule and uses energy in that process. So photosynthesis would be our anabolic reaction. This would also be an endothermic reaction, because energy is being inputted into this reaction. So photosynthesis, the creation of glucose, would be what we'd call an anabolic reaction, and also an endothermic reaction, because energy is being absorbed and a new molecule is being created. A catabolic reaction is what we're going to be looking at here in a second with digestion, right? So we're breaking down molecules to get energy from them. The way I like to remember this is like catabolic, I think like catastrophe, like things breaking apart, I guess. And anabolic, this one's a little bit less intuitive, but I always remember anabolic steroids, like, you know, guys who, you know, take steroids and they get really like, they build up really, really big muscles quickly. Those are called anabolic steroids. So I remember it that way. There isn't maybe a cuter, more succinct analogy, or not analogy, mnemonic for that. But if you think of one, let me know. I'd love to hear it. And so this brings us to the digestive system. So this is kind of a really cool process. I think we've been kind of trained to think that it's pretty gross, and maybe it is. Like, I mean, I guess it is. It's pretty gross, but it's pretty cool all the same. And it's pretty wild that this stuff happens in our bodies. We don't think about it. It just sort of happens, and it is driven by our instincts. You know, like we don't think about why we need to eat. We just know that we have to. Like, we don't think of why we need to eliminate waste from our bodies. Like when we use the bathroom, we just know that we need to. And it's really cool how the digestive system is able to just sort of keep everything going and just keep us alive without us ever even needing to consider it. And so the whole digestive system, its job is dedicated to the breaking down of food molecules to extract energy and nutrients from them. So the reactions in this component of your anatomy is designed to yield glucose. So we're going to create, well, not really creating, but we're sort of extracting glucose from it and then breaking it down into ATP ultimately. The breaking down into ATP is more the process of cellular respiration, but is sort of loosely connected to digestion. Ultimately, this consists of what we call the gastrointestinal tract and then several accessory organs. So your gastrointestinal tract is going to extend from your esophagus all the way down to the small and large intestines respectively, and then end at the rectum, and then with waste being eliminated through the anus. And so if we kind of start from the top down, the mouth is the first place that we experience any digestive process. And so, I mean, we're probably pretty familiar with this. I mean, like, it's on our face, you know, parts of it are on our face, so we can see it in the mirror. So maybe we're more familiar with this part of our anatomy than maybe other things. So in the mouth, we've got the lips, which are on the outside. We've got the tongue, teeth, hard palate, soft palate, uvula, and gums. And so all of these are super, super important for different reasons. So the mouth helps protect, or the lips help protect the teeth. They provide cushioning for that, so help guard against impact. The tongue's job is to move food around the mouth. The tonsils have a role in immune function, probably. We actually won't talk about those too much. Your hard palate's job is to sort of provide a structure and a hard surface to the mouth for the tongue to push food against and sort of break it apart. The teeth's job, of course, are to grind the food into smaller pieces, and the soft palate's job is, as well as the uvula here, the little like punching bag in the back of the throat. Their job is actually to close off this chamber in your head called the nasopharynx. And so this will eventually connect to your sinuses. So it will connect up to your nose. And so the soft palate and the uvula's job is actually to close off that opening to the nasopharynx, so that when you're swallowing food, it doesn't accidentally come out through your nose. Now, if this doesn't work, which we all, I think, intuitively know that sometimes this doesn't work, because we've seen movies where people like snort milk out of their nose, right? So there is a connection between mouth and nose, and it's that nasopharynx chamber, which will eventually connect to the sinuses. And so if the soft palate and the uvula fail to close that off, then you could wind up, getting food shot out your nose. Now it's funny, but it's also not good, because if we don't have a good way to get in there and clean it out. And so if we wind up with food or, something that doesn't belong in there in our sinuses, it's not great. How are we going to get it out? And so this could put us at risk of infection, potentially, which we certainly don't want. And so we've evolved this solution for that, to sort of block it off. And so we said that the teeth, their job is to grind food down into smaller pieces. And so the teeth actually consist of what we call the crown and the root. And so the crown is this portion that we can see. So it's everything above the gum line, where the root is going to be everything below the gum line. So the gum's job is to sort of maintain, this like tissue structure around the tooth. So it's in part helping your tooth stay stable in your mouth, but it's just tissue. So it's not, it's helping some, but it's not helping a ton. Mostly its job is to protect the bone, that, your jawbone, because your teeth are actually embedded into your jawbone with the gums surrounding it, sort of keeping it protected and in place. I guess starting with the crown, before I get a little bit too ahead of myself, starting with the crown, we have the enamel, which is the sort of shiny part of the tooth. And this is a fairly thick layer. Its job is to protect the tooth, be nice and hard so that you don't, crack your teeth every time you eat something. And also to protect the dentin of the tooth. Now the dentin, remember that anytime we see anything that ends in IN, we know we're talking about a protein. So dentin is the protein that is the main structural component of the tooth. But it's very porous. It's got a lot of like small pores and openings in it. It's kind of soft. And so we don't want that exposed to food, because things that are porous have spaces that bacteria can get into. The enamel is much, much less porous, and so there are fewer spaces that bacteria can get into. And we don't want bacteria, because if bacteria get in there, then they could potentially decompose the dentin, or they can definitely decompose the dentin, but they could also potentially decompose the enamel. But because the enamel is a little less porous, it's harder to do that. So this is why we have it protecting the dentin. Now, your roots are actually not covered in enamel. So this is actually one of the reasons why it's so important to floss your teeth and keep your gums healthy, because if bacteria get beneath the gum line, you haven't been flossing, you haven't been brushing, now bacteria are sort of starting to work their way underneath here, they can actually get to the dentin pretty easily. And if they get to the dentin, it's not a far way to go before they get to the pulp of the tooth. And the pulp is the vascularized tissue, so it's containing blood vessels, and it's also what contains the nerves of your teeth. So if we get, bacteria kind of rotting their way through the dentin, well, it's not going to be too long before you start feeling that in the nerves of your tooth, and you could wind up with a toothache. Depending how bad it is, will determine what happens next. Hopefully you just need a filling, but maybe you need to get that tooth taken out, or maybe you need to get like a root canal procedure. But moral of the story is floss your teeth, keep those gums really clean, so you can have your teeth your whole life. And so also in the mouth is a really interesting enzyme. Remember, anytime we see this ACE, that's an enzyme that we call amylase. And so its job is actually to digest carbohydrates. And so this is found in the saliva. And its job is to break them down into smaller pieces. It's starting that process of digestion early. And as the saliva and the partially chewed food mix, and it's sort of being worked around your mouth with the tongue, it forms what we call a bolus. And so this is just that like kind of ball of chewed up food. If you were to like chew your food for a little while and then spit it out and see how gross it looks, that's the bolus. And so when you swallow the food, so we've chewed, we've created a bolus, and now we're swallowing, this bolus has to first make its way past what we call the esophageal sphincter. And so a sphincter is a ring of muscular tissue that's job it is to usually stay tightly contracted and relax under specific conditions. And so the esophageal sphincter is located right about here. So it's, if we think like at sort of the end of your throat, like around where your collarbone is-ish, the esophageal sphincter is there. And so we swallow and the food moves down and it's got to open up this esophageal sphincter, this ring of muscle tissue. And then it begins working its way down the esophagus, which is connecting the throat to the stomach. And so once we make that conscious movement of swallowing, we get it past the esophageal sphincter, it continues to move down, but not voluntarily. Once it's past that point, once it's past that ring of muscle, the process that moves it towards the stomach becomes involuntary. And we call this peristalsis. And so what peristalsis is a contraction of the esophagus. So the muscle is contracting, squeezing, and then releasing, contracting, and then releasing. And when it does, it is pushing the food down towards the stomach, pushing that bolus down towards the stomach. Until eventually it's going to make its way into the stomach, where it's going to be greeted with stomach acid, full of digestive enzymes that work great at this really acidic pH. They're just living their best life and breaking down food. And as they do this stomach acid and the food is all sort of mixing and churning together to form I don't even know it's like a liquid sort of called chyme. And so chyme is what is ultimately going to be secreted into the intestines so that the nutrients can be absorbed from it. And so what you might be thinking is, how do the acidic contents of the stomach stay in the stomach and not leave to the intestines as this process is occurring? And so the reason for this is due to the lower esophageal sphincter. So once all the food is mixed up with stomach acid and that chyme is formed, now we're ready to sort of move down the intestinal tract. And so the gastrointestinal tract consists of the esophagus, the stomach, the small and large intestine, and then ending with the rectum, which is where the waste product of digestion is, or where the solid waste product of digestion is going to leave through. So right after the food has left the stomach, it's going to move to the small intestine. And so this is where we're going to have most of our nutrient absorption. So all that chyme, that food mixed with stomach acid, is going to be sort of slowly released into the small intestine, just a little bit at a time, where it's going to sort of slowly travel through and have the nutrients absorbed from it. And so the small intestine, there's a lot of intestine in a human body, but what is really impressive about it is that despite how much there is, I mean, humans are only so big. And so in order to sort of maximize the surface area that we have to absorb nutrients, evolution has given us this tool of what we call villi in the intestines. And so villi are these little like finger-like projections that are coming off of the cells of the intestines. And so these are coated in what we call the mucosa. And so think like mucus. This is like a mucus layer that is functional in many ways, but one of the main functionalities of it, besides secreting molecules that are useful for this nutrient absorption, is protecting the intestine from damage, because that chyme is mixed with stomach acid, so it's really, really acidic, and so we want to be protected from that. And so after the chyme has made its way through the small intestine, we're going to start heading through the large intestine. And so this is where the remaining waste products are going to be produced. So let me rephrase that. This is where the waste products from the nutrient absorption that occurred in the small intestine are going to be formed and sort of come together. And so one of the functions of the large intestine is that it's going to either add water if the waste product is quite dry and is having trouble moving through the intestine towards the rectum, or water is going to be absorbed if that waste product is really wet and would not be super solid coming out through the rectum. And so we call this waste product feces. It's going to be sort of created, passing through the large intestine until it eventually leaves through the rectum. We also have this little, like, kind of, it looks like a tail or like a little worm. It's really weird, as part of the large intestine. Maybe calling it a part of the large intestine is a little bit generous, but it's a little appendage sort of coming off the large intestine is what we call the appendix. And so you may have heard of it before. You may even know somebody who's had to get their appendix taken out. And so the appendix is kind of interesting because there's a lot of debate on what it actually does. There's some thought that, perhaps it plays a role in our immunity. There's other schools of thought that say, no, it's actually, just super involved with our digestive processes. And there's other people who say it has no function at all anymore, and that it is what we call a vestigial organ. I think most camps would probably agree with that to some extent, because a vestigial organ, as we've discussed before, is one that we evolved for a specific purpose, but that is no longer relevant to our current environment and the conditions that we currently live in, but that we still have. So things like a tailbone, there's a tendon in your hand that you probably can't see super well on camera, but that we've evolved as a vestigial trait. But it does contain a lot of bacteria, and that may or may not be involved in our digestive processes. And sometimes bacteria can go a little buck wild, and they can start really partying in there and growing and multiplying, and you could wind up with an infected appendix. If this happens, this is generally a medical emergency. This is generally something that you would need to go to the hospital right away. Usually, you'd know if this was a problem, you'd be in quite a lot of pain, and they may remove your appendix. It's not something that impacts quality of life afterwards. And so this is part of why it's so debated, what its role is really. And so also involved in the digestive system is these accessory organs. So accessory organs are, I think, maybe the maybe the most important part of the digestive system. So we have the salivary glands, which are located in and around the mouth. So we can see these right up here, positioned sort of behind the mouth and underneath in the jaw. And so, as we said, these are the guys who are secreting amylase as part of the saliva. So they are aiding in that first step of digestion. We also have, if we move sort of next to, above the stomach, to the left of the stomach, the liver, whose job it is to secrete a liquid called bile, which is there to emulsify the fat in our food and also process nutrients. So emulsification is when we take fat and we're able to homogeneously mix it with a non-fat. So remember that fats are non-polar and water-based liquids are polar because water is polar. And non-polar and polar things don't like to mix. This is why if you look at like salad dressing, the oil is always floating on top of the vinegar because vinegar is water-based. And so if we could add a molecule to that would emulsify the fat, then what we'd be doing is we would be making the fat into small enough particles that it can be homogeneously mixed or consistently mixed and not be separated any longer with that polar molecule. So this is the job of bile, is to emulsify that fat so it can be more evenly mixed with the chyme, so that it's not just so that you don't have any fat just like floating on top. That wouldn't do us any good because there is useful stuff that we can get from fat. We do want to be able to digest that because there is stuff that we can use. It's not all bad. So the liver is going to secrete and produce bile, but the gallbladder is this like little teeny tiny pouch just off the liver, whose job it is to store the bile. So the liver makes it, the gallbladder is storing it. And then we also have the pancreas, which is probably the most important one of all. So if here's the stomach, the pancreas is just over here. And so the pancreas is going to do a lot of different things related to digestion. So it's going to, create these enzymes that are going to digest proteins and carbohydrates, and that's awesome. It's also going to secrete a molecule called bicarbonate, which you may recognize if you're thinking like bicarbonate of soda, which is like baking soda. So bicarbonate is a molecule that neutralizes acid. And so this would be for the purpose of neutralizing like stomach acid, or maybe not neutralizing, but regulating the pH to keep that stomach acid functioning and its enzymes functioning optimally. And so the pancreas is also going to be involved with production of a hormone called insulin. So insulin's job, we'll talk a little bit more about glucose regulation in the body next time, but insulin's job is going to be to essentially modulate the levels of glucose in the blood in the body, and sort of keep everything tightly regulated so that we don't have, if we put a lot of sugar into the body, if we put a lot of glucose into the body, our blood sugar levels don't rise too high to hurt us. And also that if we start to run a little bit low, insulin is going to play a role in keeping us safe if our blood sugar levels drop a little bit too low. But we will talk more about that next time. So that is all I have for today. I know this was a little bit of a longer one. Sorry for that. And sorry also for not being able to get a video in for you on Tuesday. We will talk again Monday. I think the pattern for now is that Monday we're going to do some kind of discussion board activity, and then Tuesday, Thursday we'll do content. So we are a little bit ahead right now, which is awesome. We're going to talk a little bit more about the hormonal regulation of glucose in the body and how the pancreas works. We'll kind of zero in on that for a little bit. If you have any questions, I would love to hear them. Drop by the Q&A bulletin board and leave your questions there, or shoot me an email. Come to conference period. We haven't had a lot of attendance at the evening conference periods, so if that's something that you see yourself using in the future, let me know, because I am thinking about just keeping the daytime ones, because that seems to be what's working for people. But if you'd really like me to keep those nighttime ones because you see yourself using them in the future, just let me know and I'm happy to. And if anything else comes up, keep me posted. I will talk to you all soon.